Hello everyone, I'm Récapet. I'm an assistant professor of ecology at the University Pau Pays de l'Adour in southwestern France. And today I will present some results obtained by master students in my lab on Atlantic salmon, and more specifically on variation between individuals in the same population or between different groups of the species. So one first part will be on variation in life history strategies here in freshwater. So here what you can see is a large migrating female. So she returned from the sea and she's ready to lay eggs. She prepared a redd for that. And then beside you have a small individual that's actually a male, fully mature, but still very small, so a male pair. And that adopts a sneaking tactic. So that individual is waiting here for a larger male to come and get the female to release eggs. And then he will release his own sperm and try to fertilize part of the clutch. So this tactics doesn't require to go to the sea, but still to be able to develop large gonads and enough sperm to take part actively in reproduction. So how is it usually considered in salmon biology? Classical models for this kind of decision are based on decision thresholds. So assuming that individuals that at a particular period in spring reach a certain size or a certain mass or certain fat stores will be able to mature in the following autumn, whereas the others will rather stay longer, either to grow larger and or to migrate to sea and then reach a larger size thanks to the abundant resources in the sea, and then come back here as a migrating anadromous male. So here you can compare, that's the size of the male, that's the female, the corresponding migrating female. And then you have here the small mature pair that's able to take part in reproduction. And for example, here you have some studies that showed that there were some genetic differences in those thresholds. So for example, when the threshold was based on weight in the spring, you can see here two different families. One here at the top has a much higher threshold for maturation than the one here, meaning that for the same distribution of masses of body weight, there will be far less mature males here than here. So you have the distribution, the histograms represent the distribution of size into different environments. So you can see that here the green environment seems to be better for growth than the blue one. And that's the same for the two families. But then if you look at the density curves here, that are the genetic threshold and the error around its estimation, you can see that there is a difference between the two families, but not much differences between the two habitats. So that can be summarized like this. So the current models for salmon, they take an index that is based on reserve or fat store in a way. That's based either on a measure of structure, so it could be length, or it could be a mix of both, for example, taking body mass. Sometimes it's even more dynamic using growth rate, for example. And they use that value as the index that the crew that the organism is using to decide to mature. Well, that's convenient because that's something you can directly measure, but actually all the processes that lead to that accumulation of reserve or that growth, they remain implicit, they're not modeled. You directly take the measurement and then translate it to a threshold. And then there were different models that were developed more specifically for fish. So for completeness, I also added the model that Ben Martin was here on Monday developed, where they kind of simplified the DEB model by using only one pool for reserve and reproduction buffer. And that pool of reserve was actually used as a maturation threshold. And this is kind of similar to paper that would, for example, use fat stores as a signal for maturation. And what we try to do is actually translating these ideas into a fully grown dynamic energy budget model, where actually maturation would actually be triggered by a certain level of maturity, so of development, rather than something you can directly measure. So, which is a bit more challenging, of course, because you don't have a direct measure of that state variable. So how did we do that more specifically for salmon? So salmon has a very seasonal life cycle. So there was a first maturation decision that had to be made early in May. So whether, depending on whether the maturity threshold was reached or not, the juvenile could decide to remain juvenile or mature as a par in river. Then in summer, there was a first decision. So some individuals here that wouldn't mature could actually decide to migrate to sea. So that's here if they were large enough in terms of length, whereas the others would stay one more year in the river. And then there was also a second stratification decision for those mature pars who actually survived reproduction, which is actually a bit less likely because many of them would actually die during reproduction, and then would go to sea again. So those two tactics, going to sea or maturing in river, are not really alternative, because you can do one and then go to the sea to become an anadromous male. And then we reproduced that at the beginning of the next season. So what did we do in that particular case for salmon? We took life history data from the literature on growth, on age at different stages. And we used this data to estimate the parameters, so independently from our own data. And we got some values for the model parameters. And then we simulated growth using these parameters and environmental variable from one of the index river we follow in Brittany, so for which we have detailed temperature data, for example. And using this forcing variable and these parameters, we got some outputs that we compared to the growth and life history data from the same river for validation. So here are the environmental data. So you have the temperature curve for three subsequent years. So you can see that, for example, in 2005, the amplitudes of variation are much higher, with temperature going up to 22, 23 degrees. Whereas in other years, like 2007, the amplitude is much lower. Then for food, we didn't have that good data. So actually we extrapolated data from other rivers to have at least a good idea of what the seasonal variation could be. But then we varied it between individuals. So all the variation here in our model between individuals is environmental. It's based on food. And so either we had some individuals that got more food consistently throughout their life. So we assume that could be linked to two different feeding behaviors, for example, some being more drift feeders, whereas others would feed more on benthic species. And then we also had a look at variation that would be random between months. So one individual could have more food than the others on 1 month, but the other, it could be reversed, or any other randomly chosen level of food, which means that this allowed us to have a look at the role of variation in early life in particular, or at different stages. So here are the results we got for these 3 years. So what happens here is it's not very visible. We could have a zoom actually on these early stages, because so the one that are larger would actually the red one at the beginning, and they tend to become the mature pars later on. That's what we observe here, and that's logical given the model. But then what happens here is when they start reproducing, they invest less time in feeding. They spend less time feeding. That's the behavioral results we included in the model. And so for these organisms, plus the cost of reproduction, then we see that actually some of them had a new growth over winter or even a lower, so some kind of shrinking over winter, and they only regain growth in the spring, whereas those that were initially smaller got larger by the next autumn and went on growing during winter, which is what occurs naturally in these species. However, we are not very good at predicting the proportion of mature pairs. And actually what happens is that in some years, especially when the temperature in the spring were higher, so for example 2007, where the amplitude was lower and so early temperature were higher, we had all individuals that became mature pairs, whereas actually it was a reverse that was observed. So I will discuss that later. And that's consistent, so we got it consistently wrong for these 3 years. And then, of course, when we had random variation between months, of course, we didn't see as much amplitude, but still we had a strong early life effect. So those individuals who had higher growth early on would become a mature pair. And they would actually, within each group, the one that had more food also tended to keep higher growth later on. So then we had a closer look at prediction for growth. So here I switched to body mass to give another idea. And we were, well, when there were similar groups in a given year, we were quite able to simulate the level of body mass. So here in red, you have the values that were actually observed. And then in black, you have the values that were simulated with 2 different behavioral groups, whereas gray is just within individual variation, but variable in time. And so actually, we got quite a good median estimate for our 2 behavioral groups, although it wasn't, I mean, the decision we made in terms of the quantities of food were based on quite rough behavioral estimates. And that's true in the first fall, but that's also true in the subsequent year. Although what we didn't have is individuals that didn't mature in the second year, which occurred in the white, but we didn't have them in our simulation. And of course, it was more difficult to compare in 2007, where all our individuals matured, whereas it was a reverse in the river, all of them smoltified. So to sum up for this part, within year covariation between growth and smoltification is quite consistent with the observation, so that's good. But then growth rate tend to be underestimated in some years. So this could be because of extreme temperature events. So we saw that when the amplitude was high, for us at very high temperature, growth would be high in our model. But then actually in nature, we could have reached the tolerance of the species, and that wasn't modeled yet. We could also have effects of water flow that wasn't included in the model yet, not directly. And we could also have some kind of ecological feedback where when initial conditions are good, competition between the young pairs could be higher in the river. And so later on, that could actually negatively impact their growth. Then between year variation in life history strategies was opposite to observation. So that could be due to the fact that we didn't model very well annual variation in food availability. For now, it was the same, it was only seasonal. Then there could also be between year differences in the threshold or the time window for life history decisions. So organisms could have some kind of plasticity in terms of phenology. And then there could also be individual variation in model parameters, but between years. So different cohorts might have different parameters, but it would be a bit more difficult to test. Then quickly, I will go to a second point, which is about the effect of domestication on their parameters. So as you might know, salmon has been domesticated in a way for decades now, for several generations. So we can already speak, I think, of a domestication process where salmon are farmed in conditions that are quite far from their natural environment, very high densities, high level of parasites, but also high food abundance and no predators. And also they have been farmed with objectives that are not the same as natural selection, because the objective was to produce a large amount of meat products, so of fish meat, within the minimum amount of time. So what we did is, using data from the literature, one of my students, Valentin, estimated parameters in conjunction, as if it were, for example, multi-species estimation for farmed and wild salmon. And what we got here was, we fixed the average temperature between farmed and wild, and we also fixed the maturation threshold. So average temperature was really fixed in the model, whereas the threshold, they were considered equal between farmed and wild, but they were still estimated in the model, in the parameterization. And here is what we found for the main parameters we could estimate with our data. So we had a slightly higher, higher mobilization rate for the farmed versus wild. We also have a slightly higher allocation to soma, which is something we quite expected, because they are really bred for producing structure and not especially for reproduction. And then we also had much lower maintenance cost in the farmed versus wild salmon, and also a higher zoom factor. So as for the conductance, we didn't have very strong prediction, because of course we could have thought that in a mobilization would have been higher in farm, because to produce higher growth. But reserve is also part of the final mass, and it's not really clear whether or how the selection for composition of the meat, so the share of fat, for example, versus muscle proteins, actually was conducted, and how it could have impacted on selection of meat. And then for maintenance, here again, it wasn't straightforward, because in a way it could also be expected that farm salmon would be less efficient than wild salmon. They're not really selected for efficiency, since they are fed ad libitum all the time. But also they might have less cost. They are in an environment that is more protected in a way. They don't have as much predators. They have lots of parasites, but they are also heavily treated for them. So that might explain some of the differences if we want to interpret this. And finally, here is what it gives us in terms of growth dynamics. So we see that the growth is much quicker for farmed salmon, consistently with the higher nu and the lower cost, the lower maintenance cost. And this means that puberty is also reached earlier for farmed salmon, although the thresholds are similar. So we fixed threshold because actually genetic research in salmon tended to show that there was a strong selection for growth, but not as strong for the different thresholds. And here we do see earlier maturation, but it's mostly due to the fact that they are growing much quicker. Okay, so I would like to thank all those who contributed to the present research. So the two master students, Hélène and Valentin, some of my colleagues, Mathieu, Sylvie, who helped provide data or help parameterize the salmon model in the first place. And then also Starlight and Laure also contributed to parameterization during the previous DEB school. And of course, also people who gathered some of the data, especially the score data. And thanks for your attention.